Several international consortia have committed to sequencing and assembling the genomes of organisms across the tree of life. These ambitious projects will revolutionize biodiversity genomics, and they are possible because of recent improvements in genome sequencing technologies and assembly methods. A striking example is represented by the assembly pipeline developed by the Vertebrate Genomes Project, or VGP, who describe the challenges they face in their flagship paper published this year in Nature. Yet, the genome assembly process remains laborious and costly, requiring significant expertise, and it does not scale well to the goals of multi-species scientific efforts. To overcome these issues and further democratize the assembly process, the latest version of the VGP's assembly pipeline has been implemented in Galaxy, a well-established, public, and user-friendly computational infrastructure. Follow me to see how easy it is to use Galaxy to assemble a complex genome. Galaxy makes it easy to import data from a variety of sources, such as the Genomark AWS S3 bucket. To use the VGP assembly pipeline, simply import the workflow files into Galaxy. Customize the workflow by providing project-specific information and click Run. Once the workflow is complete, you can easily view the history and all output files. From data import to final scaffolded assembly, the Hawaiian crow genome was completed in less than 32.5 hours, with only a few minutes of hands-on time. Do you need to assemble a genome, or do you know someone who does? You are welcome to try it for yourself on Galaxy's Genome Assembly Workbench.